Tatiana Dvorkina, St. Petersburg State University, String Decomposition Problem and its Applications to Centromere Assembly and Analysis. First, I'll present a short overview of centromere structure. Then I'll introduce string decomposition problem and string decomposer algorithm. After that, I'll compare string decomposer with existing approaches, and finally show two potential applications of string decomposer in centromere studies. So why do we study centromeres? Centromere is a place where meiosis starts in cells and thus can directly affect genetic disorders. It plays the key role in chromosome segregation in all vertebrate genomes. But its sequence differs greatly from one species to another. Human centromere consists of thousands of units called high-order repeats, or HRs. The similarity between two HRs in one centromere is around 95 to 100%. Each HR represents a nested tandem repeat formed by smaller blocks called monomers. The length of each monomer is around 171 base pairs. Similarity between two monomers in one HR varies between 65 to 90%. Here we present a string decomposer algorithm that takes the set of monomers and long error-prone read and partitions this read into monomers, converting it to monoread. The accurate solution to this problem is highly important to perform centromere assembly using monomeric reads and to annotate and validate new centromere assemblies on structure level. It's also important for evolutionary studies, as it gives the opportunity to extract accurate monomers and HRs directly from error-prone reads without performing assembly. Now let's reformulate the problem more formally. Suppose we have a string R and a set of strings called blocks. We define a chain as an arbitrary concatenation of blocks, and optimal chain for R as a chain that has the highest scoring global alignment against R among all possible chains. The string decomposition problem is to find an optimal chain for R. Let's consider an example with string R and two blocks B1 and B2. Our goal is to find optimal chain of blocks B1 and B2 that covers R. In order to find optimal chain of blocks, we have to construct optimal alignment. But how to find this optimal alignment? First, let's consider a simple alignment graph for string R and block B1. It usually represented as a dynamic programming table. Each cell in this table is a vertex, and edges from each cell to its neighbors represent matches, mismatches, and indels with corresponding weights. Using alignment graphs for B1 and R and for B2 and R, we can build a string decomposition graph. We glue these two alignments graph by their zeroth row, so they have zero row in common. Label source and sink vertices and introduce new blue edges from each cell in the last row to the zeroth row. These block switching edges have zero weight and provide transitions from the end of one block alignment to the start of the next block alignment. In string decomposition graph, optimal alignment for string R and two blocks can be found as the longest path from source to the sink, and optimal chain can be directly restored from this alignment. In order to build string decomposition graph for an arbitrary number of blocks, We build alignment graphs for all blocks and string R, glue them by zeroth row, mark source and sink vertices, and add block switching edges. In order to find optimal alignment for string R and arbitrary number of blocks, we need to find the longest path from source to sink. This problem can be solved with wraparound dynamic programming algorithm. String Decomposer implements this algorithm for arbitrary number of blocks. The running time of String Decomposer algorithm is proportional to number of edges in string decomposition graph. In real data, not all centromere-related sequences can be represented as a sequence of monomers. For example, centromere X contains 6 kilobase pairs line element, and its decomposition into monomers is meaningless. String Decomposer classify alignments in such regions as unreliable and mark them with gap symbol instead of monomer ID, based on low alignment score of monomers in such regions. Another complication in real data is that errors in reads usually trigger errors in monomer identification. Again, String Decomposer tries to identify such incorrect alignments and classify them as unreliable based on small difference between scores of the best aligned monomer and second best aligned monomer to this region. So, String Decomposer usually converts the read into monomeric sequence with gap symbols inside. Although no existing tool explicitly addresses the string decomposition problem, some of them address related problems. I'm going to talk only about one of them that is publicly available, can be used on many platforms, and can work with monomers. Alpha Centauri solves a more general problem. It takes reads and tries to identify most reliable set of monomers from reads. It reports a lot of spurious monomers, and its decomposition is far from perfect. But if the set of monomers is known, as in string decomposition problem, we can use the results from the first stage of Alpha Centauri, where it partitions reads into monomeric sequences with HiTSeq, and label them with monomers. We will call this solution as Alpha Centauri, or AC, and compare String Decomposer, or SD, and Alpha Centauri, or AC. In order to compare Alpha Centauri and String Decomposer, we used Centromere X assembly version 0.7 and 12 monomer sequences from Centromere X HR. We refer to each monomer by Latin letter from A to L. We also used long nanopore reads generated by T2T consortium and extracted about 1,500 Centromere X reads that have reliable mappings on assembly by tandem mapper. We converted each read to sequence of monomers by String Decomposer and by Alpha Centauri. We also took read's origin and built its decomposition into monomers, referring to it as ideal decomposition. Further, we will consider ideal decomposition as a gold standard and compare String Decomposer and Alpha Centauri decompositions with it. We built alignments of String Decomposer and Alpha Centauri decompositions to ideal decomposition. We classified each position in such alignments as a match or error. Here, violet string represents reads decomposition, and blue string represents ideal decomposition. All blue-violet positions are considered as matches, while all red positions are considered as errors. We have summarized the number of errors for all reads for both tools. Most errors come from monomer gap mismatches, which is expected due to errors in reads. Though String Decomposer outperforms Alpha Centauri in number of errors, the total percent of errors is very low for both tools. 0.32 for String Decomposer and 0.86 for Alpha Centauri. Fewer errors allow to obtain long error-free monomeric sequences. If errors are independently distributed, then on average, String Decomposer will produce more than twice longer error-free sequence than Alpha Centauri. Longer K monomer sequences can greatly improve centromeric assembly. The number of monomer-monomer mismatches is about 100 for both tools. If we build a substitution matrix, we will see that most of them are created by substituting monomer K in assembly by monomer F in reads. All these substitutions happen in non-standard 16 monomer HRs, and there are 6 occurrences of such HRs with monomer F inside, and 6 occurrences of such HRs with monomer K inside. Further, we will call monomers on this place as FK. Then we extracted all FK nucleotide sequences in assembly and aligned them to each other and to monomers F and K. The heat map demonstrates similarities of all pairs. As you can see, there are 2 clusters. The sequences in the first cluster are very similar to each other and to monomer F, so they represent a variant of monomer F. While sequences of the second cluster are also very similar to each other, but distant both from F and K, and actually from all other monomers too. Nucleotide alignments of FK sequences from the second cluster to K and F showed that the first half of all these FK sequences is identical to monomer F, while the second half of FK is more similar to K. Thus, we conclude that FK sequences represent a novel monomer F plus K, which was created by gluing the first half of F and the second half of K. Our analysis discovered 2 chimeric monomers in centromere X, both of them occurring in non-standard HR variants. We also confirmed that a novel monomer sometimes emerges as a hybrid of existing ones. Another problem that can be solved with help of string decomposition is identification of HR repeats and their variants directly from reads. We can build decomposition into monomers for all reads in dataset and try to extract HRs and their variants by searching for repetitive regions in these decompositions. As a proof of concept, we decided to compare putative HRs that can be extracted from centromeric hi-fi reads and 26 known HRs listed in Alkan and Carter's work. Both putative HRs and known HRs were presented as a sequence of human genome monomers from the set of monomers generated in Sevim and Carter's work. From 26 known HRs, 12 have identical putative HRs. Another 5 known HRs are very similar to some putative HRs and differ only in 1 or 2 monomers. For other 9 known HRs, we couldn't find putative HRs with similar monomers. Thus, we concluded that either they are erroneous or they are not presented in this sample. Some putative HRs have very high abundance in reads and represent variants of known HRs. Have additional monomers in comparison to known HRs, or monomers not in straight order, which tells us a lot about heterogeneity nature of centromeres in corresponding chromosomes. String Decomposer is able to accurately decompose long error-prone reads into monomers, which is useful in assembly applications. Monomeric sequences obtained from String Decomposer are very important for centromere evolution studies like discovery of new monomers and HR variance extraction. I want to thank my co-authors, Andrei Bzikadze and Pavel Pevzner, for their help and great ideas, T2T Consortium team for many useful insights on centromere assembly and evolution, and to St. Petersburg State University for financial support of this work. Thank you for your time. String Decomposer is available online on GitHub, and if you want to use String Decomposer, please cite our paper.